Before we dive in, if you're in the business of building biotech tools or using AI to push science forward, check out our latest article from Ross, Why Language Itself Might Be Holding Back AI. It explores whether the very vocabulary that we use could be limiting breakthroughs in biology and beyond. You can find the link in the show notes. Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Today, we sit down with Jessica Sacher, co-founder of the Phage Directory and researcher at Stanford, to explore how data is powering the rise of phage therapy, a personalized approach to tracking antibiotic-resistant infections. We get into how phages are sourced, matched, manufactured for individual patients, the global network behind the Phage Directory, and what it takes to operationalize compassionate use therapies at scale. From wet lab workflows to computational models that predict phage-bacteria interactions, this episode is a deep dive into the future of precision antimicrobials. Here we go.
Jessica Sacher, welcome to the Data in Biotech podcast.
Hello, thanks for having me.
Awesome. Well, just to kick us off, would you give us a brief introduction to your background and what brought you here today?
Yes, so I am Canadian and I grew up in Alberta, Canada, and I found my way into a PhD in microbiology where I studied bacteriophages. And that led me, when I started in the biology of it, led me to using them for therapy in patients and helping make that possible and helping find ways of getting a personalized antimicrobial actually accessible to patients. So that's what I'm still doing today.
Wonderful. Well, for those who aren't familiar with phages or phage therapy, can you just give an introduction to what phage therapy is and the types of infections that it's used for, et cetera?
Yeah. So phages are the viruses that kill bacteria. So they're a massive class of viruses that no one talks about. And they're considered these safe viruses for us because they're extremely targeted to bacteria. And they're being used, 100 years ago, they were discovered and immediately used in humans because people found out that they actually sometimes worked. And what it is basically getting a sample of a virus, figuring out which one to use, like showing that it actually kills bacteria in a petri dish, taking that bacteria from the person who has an infection, actually growing that in the dish, and checking the virus works, and then like purifying some of it, giving it to that patient almost like an antibiotic.
Okay, interesting. And how have you seen the field evolve over the course of your work?
Yeah, it's been super interesting because this is one of those very ancient technologies that's also new, and it's had a couple of renaissances and over the course of history. But when I came into it, it was roughly 2017 when I was almost finished my PhD, started thinking about actually using phages for phage therapy. So this is like phage therapy's third renaissance that we were already on by then. And some people had started using them in patients in the US, which is where I was studying. Like there had only just been one patient, like in recent history, though. And fast forward to now, there's thousands that have been treated with phages. So just in the last 7, 8 years, it's gone from one to hundreds or maybe even thousands, even in this part of the world. So big things are changing.
Yeah, interesting. So it sounds like if I understand correctly, then phage therapy is sort of like highly personalized, both to the patient that's receiving the therapy and also to like the phage that's being developed like customized to both the bacteria and to the patient. Am I thinking about that right?
Yeah, there's like those two levels. And yeah, you have to think about, well, I think about the bacteria and the phage first because I'm just used to that as a microbiologist. I'm thinking about it on that scale. Like if you choose the wrong phage, you're just going to get no response. Nothing's going to happen. You have a Pseudomonas specific phage and an E. coli strain, nothing will happen. But then, even when you have the right phage that kills the bacteria in the petri dish, it should work. But once you put it in the body, that person has an immune system and they have a certain clinical condition. And who knows where you're putting it? Maybe you're putting it in the lung. And so now you have a lung environment to worry about. Like, does the phage work in that setting on that bug? Can I even access the bug? So, yeah, it's personalized squared, I would say.
Right. So obviously you've got lots of different bacteria that you could be targeting with these phages, and then you've got lots of different phages that you could be applying for that to those bacteria. Can you just give us a sense of like what are the orders of magnitude of the different sides of this equation?
Yeah. So, well, there's the number of phages that there are out there, and there's apparently 10 to the 31, so 1 and 31 zeros. Every time you go looking for a phage, seem to find a new one that hasn't been discovered, although that's started to plateau a little bit, but we see, so just like massive orders of magnitude of just like diversity of phages out there. And that is reflective of how diverse bacteria are out there. And people usually think of bacteria in an infection. They think like, oh, well, is it resistant to vancomycin or not? Well, they don't think about how every single bug has like, there's all the species like Pseudomonas E. coli staph, but there's also below species, there's strain. And so when staph is not going to be the same according to a phage. The phage sees so much more detail. And so I don't even know how many different strains of bacteria there would be, but I think it's the same order of magnitude as how many phages there are because they only evolve to kind of combat their prey basically.
Yeah, interesting. And so you've got sort of like this broad range of phages that can be used to target this broad range of bacteria. Can you give me a sense of why interest has grown or the number of patients that are receiving this treatment has grown over the past decades?
Yeah. So the reason basically is that our antibiotics have stopped working. And so we are seeing infectious disease doctors who are the ones called in, like they're the specialists that your GP has to refer you to because your infection's not treatable. Those people, those doctors are reaching out to find phage therapy. And that's why we're seeing a change. It used to be a patient, maybe they read about phages online and their antibiotics, they don't want to take them or whatever. But their doctor would be the gatekeeper and would say, well, no, we're not using this. We're going to use penicillin or whatever version of it we have now. But now, yeah, the problem is becoming so dire where we have just rampant infection or antibiotic resistance, and people generally know about that now, and they know about superbugs. They know we shouldn't have antibiotics in our livestock because we're just like putting them in the water supply, and so, but that just translates to like more infections that your GP has to refer you to the specialist, and the specialist is actually trying all the antibiotics they have. None of them are working, and they're saying, okay, sorry, like nothing left and maybe amputation or just like unfortunately sorry we'll just have to keep you on IV antibiotics and like hope for the best.
Yeah, interesting. And so, like, what are the context where patients are receiving phage therapy today? It's where antibiotics have been tried or not working, or there's reason to believe that antibiotics might have a negative impact on some other disease or comorbidity that's going on with the patient. Am I thinking about that right, or what are
Yep,
the contexts?
exactly. Yeah. So right now, the one thing to make clear is phages are not on the market as a drug anywhere. They are only given compassionately or in clinical trials. So compassionate, to qualify for that, you have to have had everything else fail. Like the standard of care has to have failed, and the physician has to say that we think that this will help you, but we can document to the FDA that we have tried all the antibiotics that we would try. We have done surgery if we would have, and it's still here. So the only people getting phage therapy are in that bucket where their infection is just like failed standard of care. But unfortunately, that's becoming just like so much more numerous that it's starting to feel common that like most developed countries are doing it now.
Yeah, interesting. And so you've got this problem where eventually a patient reaches a point where the doctor believes that the patient both qualifies for compassionate care and phage therapy and believes that that phages could be useful for the infectious disease that the patient is
Yeah.
dealing with. Can you just talk about the projects that you've undergone to try to meet that problem where it is?
Yeah. It's a challenging window because you have this like you have to be dire enough, but you have to be not so dire that you can't wait for phages that are need to be made just in time for each person. And so we like in 2017, that's when I met my friend at the time, now partner Jan Zhang, and he convinced me to start Phage Directory. And this was to actually address helping people find phages fast enough in that window, which is roughly like a month. maybe 3. But how do we find the people that have the phages in labs around the world, which we can get into, but it's university labs basically. And how do we connect the doctors that have the patient who have no idea where to get a phage? You cannot buy them. So you have to know someone who you're going to email. You have to look up a research paper and find the corresponding author and like email them. And maybe this paper you published in 2011, like, do you still have a phage? And would you help with a clinical case? Like, that was just not happening in 4 weeks. And not to mention the production of the phage has to happen in that time. So, Phage Directory is the main project that I've been working on since that was doing the sourcing of the phages for that. And that's kind of project 1, I guess we'll talk about.
Yeah, well, tell me about Phage Directory. So what does Phage Directory do to try to shorten that time span and meet the doctors and the patients where they are?
So, Phage Directory, we kind of basically we act as air traffic control. And so we receive requests from physicians, sometimes patients, and we tell them to go to their physician. And if they come back, which they do, and sometimes a week later, sometimes 2 years later, and they'll be like, I got my doctor. He's on board, she's on board. We will talk to that physician just via email and just say, hey, like, we'll try to reasonably gauge if they kind of know what they're getting into. Do they know about phage therapy? And are they willing to, the most important thing is, be the point person to call the FDA and get permission or the equivalent in other countries? Because we do this for any country that asks us, basically. But they always generally, if they're reaching out to us, like they know to that degree, and we will help them understand what we can do for them, which is get the message out to the labs that we have in our network. So we have about 300 labs that are subscribed to a emergency phage alert. It's a newsletter and it goes out to these labs when we've sort of just like pre-qualified a case that it's like, yeah, legit, a patient, a physician, they're going to do this properly. They just need phages. And if we do the work to find them phages, they will reasonably use those phages. Then we just send out, so we send out to the labs. We'll get emails back. Our email we send out is just kind of like, we realize we don't have to give a lot of information. We just have to give the species and the country. And so we'll say, we're looking for Burkholderia phages in the US. If you can help, email back. Tell us if you want to help by receiving the patient isolate or by sending your phages to some other lab for them to test. We realized, I mean, in the beginning, we didn't know that this would be the best way either. But this is what we've landed on. It keeps it simple. It doesn't put any liability on the labs because we're very clear that this is just the first phase that you're helping with to find phages. And then, they email us and we make the connection via email again with the physician and we'll say, hey, these are the 12 labs that have responded. We'll literally make 12 email connections. They'll get their email inbox spammed and they'll be like delighted because they never think anyone's going to help them.
Right. Yeah. So at that point, the doctor has introductions to labs that are willing to would you say, run through the potential phages that might be useful for the particular infectious disease that the patient has, and then go through a screening process. Can you just walk us through what that matching process between patient and phage looks like after the sort of connection has been made?
Yes. So, they'll sign up. They'll say, here's my address. Send me the strain. They'll get the strain in the mail and they will generally, each lab will have like, maybe they'll have 5 phages against that bug. They'll have 5 Burkholderia phages, or maybe they'll have 200. No one has 200 Burkholderia phages. Email me if I'm wrong about that. But sometimes some bugs they have lots of phages for. They will make the decision on their own how many to screen, but usually all the ones they have because they're like really want to find one. And so they will grow the bacteria on a set of petri dishes, depending on how many phages they have. They need enough space to grow a lawn of bacteria and then spot with a pipette just like one droplet of each phage they have and then incubate that overnight. And then the next day, the bacteria will grow and then there will be plaques wherever the phage was actively killing that bug and it is not able to grow. And then they'll be like, ooh, okay, this plaque corresponds to like phage A1. Which phage is that? Then they email me and say, hey, we have a phage or we have 3 phages. And that's kind of the screening process.
Interesting. And then as part of the directory, are you keeping track of which phages are impacting which bacterial strains. And so like how do you think about building up that repository, that directory of knowledge?
I wish that we were collecting that level of data. We tried in the beginning, and we do collect we collect all the who, so like which physician asked, what bug it was, and like roughly what clinical indication, like pneumonia or something, or like wound. But we also collect which labs respond and how many, we used to collect how many phages they actually actively found. But in terms of collecting like information about those phages, we kind of ran into troubles doing that. And so we don't systematically collect that. We try to keep it light so that they'll just at least help instead of putting anything in front of them like, oh, you have to have a sequenced phage or you have to submit the data because we found they just won't. They'll be like, yeah, I want to. And then it'll just get buried because they're academics. They have like a million commitments, but each one is not, it's like their priority list just drowns anything. And so we keep it light and we have not been able to capture as much as we want to. Although we have started to kind of, I mean, this is, we can dive into this, but there was a set of a couple years where we went and like collected from each lab, what at least do you care about your phages? What are you characterizing them as? We realized that was even like we interviewed a few labs, maybe like 4 or 8. We had like 400 different attributes on a list. And we're like, oh, this is like, could be a massive data set because each person cares about their specific corner of phage biology. They're like, we study receptor binding proteins. We know everything about the receptors of these phages, but we don't have a sequencer. We don't genome sequence them. No, well, we don't need that. And like another lab will be more computational and they'll know like all about certain genes that it has. Or like someone will be a formulation chemistry kind of group and they're like, oh, we know the stability and like the structure. So we don't collect that systematically for these cases, but we do at least know when we know roughly like how often we get hits, and we roughly can say what likelihood we'll find a phage for a given species at this point, I would say.
Now's maybe a good time to talk about Phage Australia. Can you just talk a little bit about the project that you underwent with Phage Australia and how that fits into the work that you've done with Phage Directory?
Yeah. So Phage Australia came about as a result of one of these cases that none of us thought would go anywhere because it was in Australia that the request came from. And they were like, we know we're far away. Like, no one's going to help us. But, like, do you have any Pseudomonas phages for this little girl who's 7, whose leg is getting amputated? Like, I don't know what the time frame was, but it was imminent. And she was in a car accident. Her bone infection just won't heal. And so we did alert, and then 12 labs answered, and this is like our biggest response yet. And we ended up finding a phage for her, and 6 weeks from the day they put out their alert, she was getting phages intravenously. And so that was like extremely rapid fire, especially for a group that had never really done this in this way before. They'd done phage therapy as part of a clinical trial years back, but like to do the sourcing and like get the paperwork to do the compassionate use and everything, like all that had to happen in that window. The phage that they found came from Israel, got purified in the US, got sent to Australia for use. And like, so it was crazy. That was in 2019, fall of 2019. So we were so excited that that worked so well. They were like, yeah, we actually have a grant probably that we're going to be able to get to build out something like bigger, and like basically scale this up to more patients. And the Australian government was really excited about that. And they got that grant. They were gonna they offered to just bring Jan and I out there to like join their team hands on, like actually be facilitating them building out their phage therapy ecosystem around the country. And so that was extremely cool. And we were like, well, we've just been working on this remotely from our couches. This is before the pandemic. We were on Zoom every day. Like it was weird. But so we were so excited about that. Pandemic hits, froze the grant. And unfortunately, like 2 years passed where we had to kind of like roughly work on this remotely with them. But we finally got to Australia in 2022, early 2022. And yeah, then we were there. And I thought when we got there that like we knew, so Jan's like the data guy, like data engineer and software and user experience. So he was going to build the data, everything for them. Like, what are we collecting? How are we going to make sure it's usable and learn from it? And then for me, I thought I was going to be doing like external, like trying to get us more sources of phages, make sure that everything's working. But I quickly realized that like I was going to be in the lab because there needed to be somebody to actually make the phages. And they wanted to scale that part out, really. Like they didn't want to keep having to hope that they would find someone in the nick of time. Like that was kind of what the funding is for. That was really cool because after a few years, I think 4 years out of the lab, then I was back in and trying to now make my first, like literally first probably month on the job. Like the physician who I was working for, John Iredell, he's like, okay, we have a patient next door, my patient, because he's a physician and he's running the lab. He's like, so he needs phages. Like, could we like make him some? Do you think? And I was like, well, yeah, of course. Like, yes, that's the goal. He's like, so like this week? Like, when? Can you just start? And so that was kind of the big eye-opening moment where I was like, oh my gosh, okay, I'm now gonna make the phages. And from this point, like be the boots on the ground version of what I've been like helping orchestrate. So that was really cool.
Yeah, so you were sort of in the heart of it, like making the phage development process operate more efficiently. Can you talk a little bit about like what you learned about the operationalization of phage therapy from that experience?
So I just like cherry picked my favorite protocols from the different labs that I have been seeing and working with, and then strung that together in a circle, drew it on it, literally drew it. And I had another postdoc in the lab, Stephanie Lynch, who she was hired at the same time, and she'd actually been a Phage Directory volunteer, and I like got her out with us. And so we were like, okay, we can build this from scratch, and everybody's going to listen to us, and we're going to try to hopefully like check all the boxes on the regulatory side, but we're going to do what we want to do in the lab. And so what it ended up looking like is there's a diagnostic phase where we would bring in the strain from the clinic, and we had to work out how are we going to do that from a communication side, but how are we going to make sure like we are keeping track of the pseudomonas that comes in today is not the pseudomonas that comes in like tomorrow. Very critical point, but very overlooked. And then screen our phages. Okay, we're going to screen. We had to find which phages do we even have. We found a bunch in the lab from decades of academics prior, and we kind of like brought them in together and started to gradually like go through them and see, are they all unique? Are they sequenced? Are they mixed? Start to create a bank. Then we would, in our process, we would select, we decided like 3 to 5 that showed a hit. We would do a subsequent set of experiments to like feel better about them probably working in the patient. So instead of just a spot assay, we call it, where you pipette the droplet, you would do like dilute that down, serially dilute the phage down, because it's always like orders of magnitude that the brain does not process, but like 10 to the 11 phages per mil are in your tube. So you have to dilute it down kind of like 10 to the 11 times so that you can actually see one plaque. And once you can see one, it's really a phage, and it's not just like the cells died from impact of like a lot of stuff hitting them. Like if you see like single countable plaques that like go up in number as you dilute, they that gives you the sense that that's really replication and that's really happening. And then we would also pit them against each other. We had a step where if we had like 3 phages that look good at that stage, then we would like combine them in each combination and do a growth curve. So we would see if the bacteria like they have their growth curve and see if they are inhibited from growth with all those combinations, or maybe 2 phages cancel each other out. We didn't see that a lot, but sometimes. So things like that. And then that was the diagnostic phase. And so we would put together a report and came up with what the report should have. That would be for the clinician to say, okay, this is why we think you should use these 3 phages. Like, here's the growth curves with them. Here's how they work in combination. And yeah, and then we would have, so it's pretty minimal though, pretty minimal information that they really needed it then. And then the production phase was after that. And so Stephanie, we eventually bifurcated like roles or specialized. She was the diagnostic person, I was the production person. So she would give me the 3 phages and say, okay, make these. And so then I would produce like a larger batch of each of them and filter out the bacterial debris because they lyse the bacteria. And that's the most toxic part of the process. And that's definitely something that would harm somebody if you didn't clean it out, because that's our immune system. It's like the innate immune system, even like it's very primed to find bits of bacterial cell wall and get really mad about it. And so you filter all that out, and then we figured out like putting through the phage through a purification column, kind of like when you purify any protein for people to do that. The same machine, like FPLC, Acta, Pure, whatever you have, you can do that with phages. And so we would do that to just purify it more and remove all the debris. And then we would have the phage, it would be now produced, but we would have to do like quality control and formulation. And so this is the last kind of two steps. So the formulation and quality control happens all through, but we have to decide like what is good enough, like what is safe enough for it to move forward to the patient. And so there will be a sterility test. So we would like make sure there's nothing growing in there. Not, like if you plate out and grow in a blood culture bottle, you're not going to have any bacteria in your solution. Then we would have like an endotoxin test, which is the measure of that cell wall and things like that. So we put together like a set of 5 safety checks that we would do. And then we would formulate it. Basically, we wanted the pharmacists to do this, but they were not quite ready. So we would have to actually like source glass vials with the little needle plug, the rubber stopper, and we would dilute the phage to the concentration that we're going to treat at, and then distribute it like with a pipette, like 1 ml into each vial, and like put those in a little box and print off a label from Microsoft Word and like tape it on and be like, here is the product. And we would walk it in a little organ, what are they called? Cooler through the hospital and give it to the nurse's station. So that was the process. Yeah, that we put together.
It's just an entire pharmaceutical pipeline in a box,
Yeah,
which
just compressed.
just compressed. So obviously, this podcast is called Data and Biotech. So you've outlined this. You've outlined this process. You've got this process that you created from the best protocols that you could find from the labs around the world, and you've got an established process that's working for you. But obviously, your goal is to treat, and the goal more broadly is to just treat more patients, or treat the same number of patients at lower cost, and or treat the same number of patients with fewer diverse things that need to be created, just less effort. So how do you think about, or how did you think about that in the context of Phage Australia, and then how do you think about that in the context of where phage therapy goes from here?
Yeah, it's interesting because like I just like at the beginning went really heads down and just like, okay, we just have to do this for one patient because there's one guy across the hall, and there really was. And so we put all of our effort on that one person, and it took us 3 months, and we treated him, and he had a great outcome. And like then we were like, as soon as that happened, we're like, okay, how do we do this? I mean, faster. What would be the best way to speed it up? And we're always getting these questions from like the bosses and the funders, and so we're like, well, I mean, we're just two microbiology PhDs. Like we're like, well, you could give us more people, but that wouldn't really speed us up. You could, I mean, we could make a bigger batch. They would always ask us that, but that doesn't help either because our small batch already makes 4,000 doses, and we, we're limited so much more on like we wouldn't find 4,000 patients who need this phage. So there's no point in scaling up the batch. And we sort of just like organically fell into like these ways instead of just being like, how do we scale this? We just started realizing like what would actually save us steps and then work from there. And so one thing that happened was the the second patient that came in that needed the same phage we'd already made a whole workup for. We'd done their phage like safety screening assessment, and we had it already like made. We were like, oh my gosh, we can treat this person next week. We only have to do the final QC again. And I like that's obvious to someone who's used to thinking about data, scaling, and like process, but we're just microbiologists. So we're like, whoa, that's what they mean by scaling. One thing that would be useful is if we had like a larger bank of phages, like vastly larger. We only had about 300 phages. We were able to find phages against each patient that came in, but it still took us like that week of screening for each. We pretty much allowed a week for each diagnostic. So obviously, like you could, if you had a larger set of phages, you would more likely be able to find one. So you either have to have a lot of phages on the shelf, a lot, or you have to just like get really, really fast at making them. I don't think you could press that too much. And like, I think one big blocker is like, so we've talked about this, but the in silico part of it, like, can you predict the phage without even doing any testing? Like, in theory, yes, if we had enough genome sequences of the bacterial strain and enough genome sequences of the phage, that we put them all together and we collect that data set. That is like something that could shrink that diagnostic window down. And sure, you could get it from a week of work to a click. And that would, for sure, that would save like a week per patient. So I think you can start, there's just like a lot of low hanging fruit that you could start to do. It's just a matter of what is somebody motivated enough to kind of like decide that they're going to. Like, how do we get 10 patients treated in a week instead of one? And I would say the other huge thing is like, this is such a local enterprise almost. It's like the patient kind of needs to be next door. And even if we had the phage on the shelf, like how many patients are we going to have next door? Like you kind of have to have this set up in multiple places to be able to scale it to all the patients and all the next doors. So I think about this a lot, but I quickly realized that there's just like chunks of things you could do here, there, and everywhere. And ultimately, yeah, you could like scale it in the way that similar to like the CAR T cell therapy and like how you could have autologous one or you could have donors that and like, okay, 10 people can get that. I think that's the way that it goes. Or fecal transplants, like you start getting like awesome star donors and they just donate all of it. And then you have the safety down and like that part is fixed. I'm like, okay, you can help 80% of people, not 100%, but that's fine. It's very much I think that's the way to think about phage. The other piece I might think of is kind of the communication piece and just how you even make this something that's an option for all the clinicians around the world. Like, I'm sure that other fields have had to deal with this too. But even if you have this like beautiful system that like, yeah, if people find us, we can help them, even if they're not next door. But it's not part of the system. Like the hospital pharmacy does not think that that's one of the options in their arsenal. And so I guess I try to find a place for the communication piece because I think that's a huge way to scale it too. Is like once there's awareness and it's not this like mountain of like work that this one physician is responsible for, I think getting that to be just more at their fingertips could actually really move the wheel.
I'm really excited to have you on here because I feel like all of the problems that you're facing are sort of a microcosm of the biotech landscape at large. And so I think that this is just a really fun example of the places that data intersects biotech. So with that as an intro, can we talk about the two papers you shared and sort of the data science methods that are being used for phage therapy?
Yeah, so I have gone through a rabbit hole recently and looking at like, are people getting close to this whole like in silico prediction? Like I kind of people used to consider it, and I still kind of do, like the holy grail of phage therapy is like being able to predict it from genome alone what phage to use. And people have tried to do this over the course of time since I've been in the seat of Phage Directory, like seeing it. There's lots of little efforts to do machine learning with phages, and like people will go and collect a bunch of get public data, maybe phage genomes, and then they'll try to maybe they'll do an experiment where they'll pit them against a panel of strains, and then they'll kind of come up with some matrix of like did it kill or not, or like maybe a 1, 2, 3 score, and then try to kind of use whatever model is available like at the time or build whatever they're going to build. And obviously that's shifting now with all the new LLMs and everything, and like way more activity in the space. So my sense is they have a lot more options on the tool side. But so like there's been these efforts and I kind of went down this rabbit hole last summer, I would say, to figure out like where are we at? Because you hear about it and then yet we're not using it at all. Like none of the clinical efforts that I'm using working with are actually saying, oh well, let me just predict like we can get the sequence of the bacterial cell and feed it into something. Like nobody's doing that. And yet I get the sense that they're doing it in research labs around. So why is that? So I found these two papers. One of them is like they're both kind of very similar, and it's kind of a cool comparison to look at it because they're doing strain level phage prediction, but they're each shows a different species. So one is E. coli and one is Klebsiella, and they're using different ways of doing it on the machine learning side, and that's the part where I'm like, okay, I'm not gonna be able to assess like XG boost is better than this one. Like, okay, I get that there's a lot of ways that you can like check what is going to like be predictive for you of what your lab results say. But it's interesting looking at the data collection side for me because what they're taking, first of all, I thought it was interesting like they're not I would have thought you needed thousands and tens of thousands of each interaction to ever make a model, because that's what I had in my head as like the case for machine learning. Oh, well, you'll never do it. It's all about the data. And of course it is. But they're doing it with like 100 phages, 100 strains, or 400 strains, 100 phages. And so you get like 10,000 or 40,000 interactions. And that is enough for them to like be reasonably like predictive, like over 80% or whatever I think each of them are, of choosing a phage cocktail that's going to work reasonably well, or like ranking the phages that you should test in order of like what will likely give you a hit, and like having mostly those that ranked list like be something that does contain real hits in the lab. So that was cool. So they're number one, people are doing strain level phage prediction, and they're doing it with on the order of hundreds of phages and strains, like low hundred on each side. And so I think that's really cool that we're kind of getting closer to something actually feasible. And I bet a lot of these phage therapy centers that I work with now, they all have that amount of phage and strain. Like that's like within the order of magnitude that most labs would have. And so they could all be training their own model and following the exact same methods in these two papers. And I'm like, wouldn't that be cool if like, because I think it probably is very important that or it's probably not generalizable, and obviously they can only to some degree test that, but it's probably like for this set of phages, like here's the model, or for this set of strains. So we probably need to like really figure out how far that goes, or like but but it's like tangible enough that one lab worth of information could actually meaningfully do that. So I think that's what I'm really excited about, I guess, for those papers.
Yeah. So my takeaway from these papers was sort of similar to yours, which is that what we validated is that it is knowable to a certain degree from the genetic code of the phages and the genetic code of the binding proteins that you can characterize the host range for a given phage within a given set of bacteria, and you can have a reasonable degree of accuracy that even if it doesn't tell you in silico this is the phage that you need to use for this particular patient, it could at least get you much closer to these are the phages that you should be testing so that you can close the loop between patient and phage much faster.
And just to like piggyback on that, like figuring out like should we send the strain to all these 12 labs, or should we just work with that one in Israel because they have like 3 of like the 10 hits basically, or like put their library in and just know like yes or no to even that entire effort. I think that's yeah.
Yeah. I mean, the other thing that really jumped out at me, I think it was the Bocarts et al. Paper, and we'll post the two papers in the show notes here so that everybody can go and look at the papers. But I believe they used ESM2, the embeddings from ESM2 to get, even though they're only testing like thousands of interactions between phages and bacteria, they're able to get such much richer information about the nature of the phages and the nature of the bacteria because these large protein foundation models have been trained that incorporate all of these different types of data that, even beyond sort of the genome of the phage or the genome of the bacteria, can give you a lot more signal about how these proteins are likely to interact with each other. And so that to me was enough. In addition to XGBoost has been around for decades, like the nature of the methods being applied to the problem are relatively simple, but the amount of information that can be brought into the equation from these foundation models that have been trained is just really improving the ability of these models to pick up on where a phage host match is likely to occur. Am I
Yeah.
thinking about that right?
Yeah, yeah, and I think it's so cool how, like, it almost goes backwards to the biology and, like, based on what factors ended up driving what was chosen as the phages, they can kind of and based on what they reported, like, made the model work. It's like, "You know what? We didn't need the whole genome. We only needed this set of genes from the phage and this set of genes from the host." And, of course, like, I think that's a limit too, but it's also an opportunity, but, like, you have this, like, information showing you that, oh, okay, those are the drivers of phage host interactions in this species. Like we kind of thought they were. That's why they chose to extract those. I was like, it was an informed phage bioinformatician who decided that, and like a regular person wouldn't. You'd have to know something about like the phage host interactions to even choose like which parts to pull out, but it also kind of helps you validate, like, if the model works in the end, like, yeah, you were right about choosing those and like receptor binding protein and like capsule genes for the host. Like, was that the thing driving it? Like, yes. And the E. coli paper, like that group, I mean, I haven't talked to that group. I know some of them, but I imagine it was kind of surprising for them because I know that they're a group that came from phage defense genes, like looking at like CRISPRs and all the different versions of that that are coming out. And that's like a hot area in phage. But what they showed is that that part didn't actually drive anything. Not nothing, but like they reported actually defense isn't what's driving phage host interactions. And they have to report that as like, but I imagine like that's what they've been studying. Obviously they think that's important. So you kind of get this information just by even trying to build these models about the biology. I think that I love that. Yeah, and I can get a lot of other phage people excited on that side because sometimes they're like, "Well, we don't need the machines," but it's like, oh, the machines are just so they're going to give us lots of rich things to test.
What's unique, or what have you learned from sitting at the intersection of wet lab and computational opportunity, and what are sort of the challenges that you see in being in that position?
Yeah, yeah, that has been very interesting, and I so from the beginning of like my beginnings were not I wasn't in a computational lab. I was like very much wet lab. And met Jan, he was like, "Hey, computers can help your field." And I'm like, "Well, the computer people can just make websites. Wow, now we can actually get our pages to people." And like slowly and I've been dragged forward toward like what the computational side can do, but I've always been like there's never been enough time for me to like go and learn bioinformatics and learn it all myself and figure out what databases are. I'm like trying constantly. Like that has been so the way to characterize my last like most of a decade is like me trying to catch up. Like my biology knowledge is always feeling relevant, but it's like always like I have to interface with the computational people or there's no time for me to become that. And so it becomes really interesting. And I at Phage Australia, there was a bioinformatician on our team. There was Jan, who was the data engineer. And I before that would have thought like, oh, you just bioinformatics, you take your phage genome and we're gonna see like, is it safe? Does it have any red flag genes? And are we gonna use it? And which phage is it? And I realized like, oh, every time she gets a new phage, she has to like develop a new pipeline for that and new set of steps because they're each so different from each other that it's not just like computer click done, which I think a lot of us come at this thinking and all these black boxes, but we always just have like a weird amount of like trust that the computer can do it, but also like no trust because like how? But I I've gotten way more my grip on like to what extent it's like a massive whole world of complexity, even for the computer people. Like you're not just gonna like replace something with it. You're like, you actually now have to be the one choosing which tool when. And if it's an E. coli phage, I have to use the tools that are working for E. coli phages. And then if it's got a lot of repeats in its genome, you can't use this tool because it will be sensitive to that. And it's just like I just realized it's not that different from like my biology training because there's all these rules and there's all these contexts and each bug is different and the growth phase will impact things and like oh it's been so cool and interesting, and but interacting with the actual computational people, that's the other thing is like, so I live with one, and he's like, the questions are like, why does your field do it this way, or like, why do you guys not even have this data collected? Well, when you're in the lab and your gloves are on, like, you're not able to type on a computer. And so you're not capturing data. Like, the problems are like, as you start working with the computational people, you realize like the problems are not like how to compute the thing at all. They're like how to make computers see what we see in the lab in time. And it's more about like people logistics and physics on the level of like I physically can't capture that data in real time very well, or like I don't have ugh, even thinking in frameworks, doing things in the same way every time. Like biologists, like I think we're just so there's so many, and not just biologists, but like anyone in the lab, anyone cooking, to document exactly what you're going to do and to do one thing the same way every time. If like you're not going to, unless like why? Like it's so hard and it doesn't really give you anything. So I think addressing like the problem just shifts way more toward me, the biologist. And I think that we have like to have a seat at the conversation when these, like, say a database is going to be designed to collect all the phage data, like Australia. Okay, we wanted to do that. We try to do that. Jan's like, okay, I'm going to make the database. What data is important, and when do you get it? And we're like, we can't answer it. And like, well, it depends. I don't know. And so he has to follow us around, and he's like, what? Your bug is shifting every time you grow it. So what do we put in the database as like its identity? It's like, well, it kind of doesn't have an identity. It has attributes. And how do we know that it hasn't drifted genetically? Like, well, we sequence it. Let's store the sequence data. But when do we need that? And how often do we sequence? Because technically it should be like every time we use it. But we can't do that. So I guess in a long-winded way, it's just like been interesting to realize what the computer people need and like how much I can actually contribute just by explaining like the day to day. But also, even with 7 years of doing this, and even like living with the person like they're building the data infrastructure, and I'm doing the lab work, and I have freedom to decide what I'm going to do, and he has complete freedom to decide what he's going to do. It's still like extremely challenging. So like anyone who wants to kind of make that easier, think about like how do we capture information from a lab? That is like still very much, I think, like where the problems are. But
Yeah, I mean, you touched on a lot of themes that we've had in this podcast before, where there are lots of people building software for biology to try to capture data more effectively, to take advantage of the of lab automation, which then makes it so that biologists no longer have to hand code the data that's coming off the line. And also in the machine learning world, there's sort of the bitter lesson that everyone cites as you shouldn't be you shouldn't be making rules about your data, about which data is valid in which circumstances and which methods need to be applied and in what contexts. You should just be generating as much data as you can and relying on the data to tell you the story, which when you're talking about human generated data at internet scale, I think makes a lot of sense. But when you're talking about in the context of biological data at biological scale, where the numbers of what we can measure and the quality of the measurements that we can can't get quite as large or with as high a degree of resolution, we're in this interesting place at the intersection of wet lab and dry lab, where within like sandboxed environments of like new fully automated biotech organizations, all of that data can get generated and you can rely on the data to tell you the story. But in the context of therapies like phage therapy that are still under development and where the processes aren't automated or very well defined and where the business model hasn't been created yet, it's unclear how these data science principles necessarily apply.
yeah, totally. I think you just have to like be seeing it as the whole set of steps, like your whole pipeline, and some things are going to be like great for automation or great for like a anytime you can like stick in a tool like, okay, the phage genome is assembled, put it through this thing to find toxin genes, like for sure use that and then move on. But for like anything with just like be allowing that not your entire process has to be like perfectly automated, or like that the data collection is just going to be there. You could have like wide swaths of the pathway that are just like, you know what? No, we're not going to, we have not, this is not standardized as much. That's okay. Like draw a circle around that. But like this part, this tiny little part is like actually just an algorithm. Like which of these three phages, like what I described of the growth curves, like we could like take that and like quickly pick which one to use and modularizing it. And I think we can get a long way.
Yeah, that's interesting. And I think the problems you've described are sort of the problems of startup biotechs across the ecosystem, of figuring out which of the aspects that we can standardize, that we should be
Yeah.
standardizing in order to capture the data that we need, versus which of the aspects that need to be by their nature flexible until we've determined what the therapeutic model is that actually
Yeah.
solves the problem we're trying to solve. Well, this has been a really fascinating conversation. And as we look toward the end, I just love for you to share what are the different directions that you see phage therapy going in the future, the next 5 years, 10 years?
Yeah. One thing we've seen emerge is these sort of phage therapy centers, we call them. And they didn't start, used to call them pre-phage therapy centers because they weren't really a center. They weren't calling themselves that. But they were like a place that was like repeatedly doing like compassionate use case production. Even if it's just a research lab and they have like one person who like does this on the side, you kind of start to see them as like, okay, they've got a little process and they're now a center. And so these have like grown up in the last 5 years, and now there's different ones that kind of are everywhere. And that I think that's going to be here to stay. And I think like these academic medical centers are going to keep having to be dealing with like for now all the infections, but eventually like the long tail, even if we have phage cocktails that actually make it to market, which is happening gradually. So I think the future is a combination of phage, like a pill that has 3 phages in it for Pseudomonas, and you use it for this like reason because you have a Pseudomonas infection. Like we'll have species level cocktails that will come on the market as a biologic. That has been so has been t- it's just taken a long time, but it's like we just had a successful phase 2 trial last week announced from Biomics. So that's now only the second positive phase 2 phage therapy trial ever. And so that's a really exciting thing. And I think it'll get there. But the money in antibiotics is so scarce. It's just like the worst choice for a business that you can have, like max regulatory burden barrier and like pretty much no sales volume because it cures the thing in a week and it's like considered cheap. So, you're not going to command a high price for it. So it's like I don't really think that the phage cocktails on the market are going to be the thing that like saves us, I guess, because it's just so expensive to get there. And so that's why I think these phage therapy centers are just going to keep getting a little bit more scaled up. They're going to like do their local region. We saw the Australian one that we worked on while we were there in the 2 years we were there. People were like, okay, we're starting this in Canada, Singapore, Nepal. We had like contingents of researchers and physicians like visit us physically in Sydney. And they were like, okay, we need to know what do we need to do. So I think that's going to keep expanding. There's one in the UK, like there's like multiple in the US. And I think that'll support people in the next 10 years. And it's just going to keep going. But as long as the problem is the way it is, which is the case, like superbugs and the whole crisis, like unless we fix the economics of antimicrobial development, which I hope we do, I hope we carve out something that puts them in their own category, basically, and says, you know what? For these, they do not have the same economics as a drug. They will not be developed if we make them cost the exact same amount to make. So, like, if we did change that, that would help. Maybe phase wouldn't be as needed.
Yeah, so I listened to. So for listeners out there, if you're interested in phage therapy, you should absolutely follow the Potavirus podcast that Jessica hosts. I remember one of the episodes I listened to talked about, I think it's the Pasteur Act, and the idea that the government would would sort of be paying on a subscription basis for access to phage therapies rather than having the economic model be based on the sort of the same method that's being used to reimburse for antibiotics. And so there's like these creative ways that we can come up with within the context of healthcare systems to make sure that these therapies that do have real benefits are being economically supported.
Yeah. Yeah. I think like for beyond antimicrobials, like and phage, like as I've come to think about this, we should want a diversity of like types of ways drugs are made, because otherwise, like the only drugs we get are the ones that happen to fit this like volume-based reimbursement model. And I used to be like, whoa, what are all these jargon? Like I, as a biologist, didn't care about that. But it's like, oh, it makes sense. Like if you only can make drugs that meet a certain, say we could only have businesses, but if you sold 10 of them, then you get the money. But like you can't have insurance businesses where you kind of get money anyway, but you help them if they need it. Like we need to kind of diversify the models, and then we'll like diversify the types of treatments that fit. Because something like phage just really doesn't fit the current model, and even an antibiotic doesn't fit it. And so it's like we can't have nice things because we haven't been creative about creating the structure. So I think there's like tons of exciting stuff to do on that side of things.
Awesome. So as we head toward the end, is what's your work at Stanford looking like, building on the stuff that you did in Australia?
Yeah, so that is, yeah, exciting. I came here last year, middle of last year, and we're working on developing, as I mentioned, like on the phage engineering side, like what can we do with using phages to deliver genes and deliver peptides? Like phages have been used as phage display in the drug development world for forever and ever. But what about like therapeutic phage display in a sense? Like we know they're very engineerable and we can get them into eukaryotic cells. This is kind of a new-ish frontier for phages. Like thinking about their use beyond just like bacterial acute infection, I think is like really gonna it, it makes sense with the times because no one wants to fund acute infection treatment. But also it's like legitimately you have this very engineerable entity that is very pretty innocuous in the body from an immune perspective. And also I think all these foundation models, back to the Evo thing and everything, I'm noticing like the biosafety like rules on them are like, do not use human viruses because someone might design a bad virus and we'll have COVID again. And so phages are allowed to be used in these models. And so these models are going to get good at making new phages way faster, and they're going to get good at making like new AAVs or new other human viruses. So I feel like we should push on that. And that's what I'm excited. We're going to kind of try and build out like a phage delivery center in a sense, or pipeline here. And Stanford's very close to all these different groups that want to, Lab A has a big CRISPR system they want to deliver and they can't. And Lab B has like CF genes that they want to deliver and they can't. Or there's like a cancer lab that wants to deliver a peptide that will like stop the cell cycle from being messed up. And so we're going to try and position ourselves and just like see what it takes for each of these problems to like, can we use phages to like get the thing where it needs to be? But it's like a whole new frontier, I would say.
Awesome. And where can people find out about your work and connect with you online?
Yeah, I'm on Twitter mostly, Jessica Sacher at Jessica Sacher, and you can find me on LinkedIn. Phage.directory is our Phage Directory website if you're interested in that. But yeah.
Right. And people should definitely follow the Potavirus podcast and the
yes.
Capsid and Tail newsletter as well if you're interested in phage therapy.
Thank you.
Jessica's on so many platforms and sharing so much information about phage therapy that even she forgot some of her platforms. So
Yeah,
Jessica, it's been a pleasure having you on the podcast. Really appreciate the time and look forward to connecting down the line.
Thanks, Ross. So fun. So excited for this.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.